Below the Red Sea, a coral reef is investigated by a group of young biologists from Cambridge, England.
Trio people.
Coral is not just a rock. It's an animal, a whole family of different animals who create a fantastic diversity of forms and colors. It's a live, thin surface layer that coats the dead rock that is the mortal remains of countless earlier generations of coral animals. At first sight, this appears to be a marvelous example of the balance of nature, where harmony lives with beauty, and all is founded on the one essential, coral. The Crown of Thorns starfish has been cast as the villain of the story. 10 or 15 inches across, sometimes as much as 24 inches, it's covered with vicious-looking spines. And it's unique in the damage that it can do, for it eats coral. It can devastate a reef, destroy the balance. These corals are dead, the animal life that created their fantastic forms digested out of them by the starfish. In Australia, vast hordes of starfish have been reported as killing large areas of the Great Barrier Reef. Around many of these islands, most of the coral is dead. Some scientists claim that the truth has been hidden and their data ignored. To some, it's all part of a world ecological crisis. Others, no less qualified, disagree. In Australia, scientific investigation has been overshadowed by overstatement. Amid bitter controversy, it's become difficult to decide on public policy. The Crown of Thorns has become a political issue, and it's also a problem for the Australian tourist industry. Dr. Peter Vine.
On Green Island, for instance, I knew a tourist operator there who had previously been taking tourists out to see beautiful coral through a glass-bottom boat. And when I was there, he was taking them out to see the Crown of Thorns starfish eating up all the coral. And now I suppose he's taking them out to see the seaweed where the coral has been eaten, because there certainly isn't any beautiful coral there now.
On the Great Barrier Reef, the starfish has been studied in plague conditions. Further dramatic and at times emotional reports of other plagues have come from American scientists working from Guam, where native fisheries have been damaged. A third group has been a British team working in the Red Sea. To try to discover the truth about the plagues, the Cambridge Coral Starfish Research Group came here in 1968. Its organizer, historian Dr. Christopher Rhodes.
We came here to Port Sudan when we decided there was a question which had to be answered because it was the nearest place to Europe. We looked at the map, literally, to see where we could get to on the money we had available. And we also had in our minds very much the fact that the whole situation was a plague one in Australia, and that such research as would be done in Australia would be done under plague conditions. And what we wanted was a state of balance. And to the best of our knowledge then, and we asked a lot of people, here, around Port Sudan, we were going to find a state of balance.
This program is the story of this single concentrated study that's been going on now for over 5 years. It includes their own filmed record of the work that they've done, the expeditions, the investigations, their occasional mistake, and also their success. They built themselves a platform on a reef 12 miles out from Port Sudan, with scaffolding shipped from England. Occupied night and day, it's now a permanent center for the continuous study of one special reef. There's constant activity in ferrying out fresh water, fuel for the generators, and for the air compressor that's needed for aqualungs, bits of equipment, biological specimens. And with them here, a new member of the team who's just arrived today. He inherits the results of 5 summer expeditions, of which that in 1972 built the platform. After a long period of planning, it was put together in just 2 days and equipped with camp beds, a cooking stove, and a little shade. As a finishing touch, someone had the idea of providing it with a windmill to generate silent electrical power. It lasted 1 day, It inflicted several minor injuries and might have shaken the whole structure to pieces. Sadly and more noisily, they now use a generator. The platform stands in about 4 feet of water, a depth that varies little the year round. There's not much tide here and no storms. Harvey Reef, about half a mile long by 100 yards wide, is just one of many that fringe the Sudanese coastline. This almost island is the home from home of Rupert Ormond, the scientific leader of the group, who may be living here full-time or simply visiting the platform by day. None of the team disguises the fact that it's a very pleasant, if rather unusual, way of life. You don't wash up, you just throw the dishes in the sea. All around the platform are the regular inhabitants of a coral reef, scavengers who are only too happy to help pick the plates clean. On this reef, man is just another aquatic animal, and to the creatures around him, one that seems pretty harmless. His aim is to be accepted, but to disturb the natural course of events as little as possible. Over a lunch of army field rations, they discuss current research into ways of dealing with the Crown of Thorns. But in such a complex environment, before you can take effective action, you need to know as much as possible about the animal itself. When they started work 5 years ago, very little was known about it anywhere in the world. So here it is, the first object of their study. Under its pincushion of thorns, it can live for 8 or more years. The number of arms, generally 13 to 16, varies from one animal to the next. So too does colour. On coral, this would blend into the background. Here the arms are lifted to show the crown of thorns feeding on a piece of coral. Through a mouth in the center of the underside, the stomach has been turned inside out to cover the coral and bathe it in a digestive juice which simply dissolves out the living tissue of the coral. And it holds on with these innumerable little suckers that are called tube feet. The scientific name for this animal is Acanthaster plankai. It was first described in the 18th century, but only recently has it been shown to be so very widespread. It's not an easy animal to find, and with so much else to look at on a coral reef, this may have been regarded with more wariness than real interest.
The spines are very sharp. There does appear to be a small amount of toxic material on the outside of the spine. They do in fact have a semi-crystalline tip on the end, and this penetrates very deeply into the finger if you just happen to put your finger in the wrong place. Now the result of this is that if you get just 1 or 2 spines in a finger, you may just get a small swelling following from this prick, but nothing really uncomfortable. On the other hand, if you drop one on your foot or stand on one, then I think you'll probably be very ill for a couple of days. But certainly there isn't any specialized poison system, such as you often get with sea urchins, where you get the whole spine filled in its hollow cavity running down its center with a very toxic venom.
And here's another of its defenses. It generally keeps out of sight by day. And when brought out, its immediate reaction is to roll itself into a ball like a hedgehog. In fact, it's not too difficult to handle like this, even with the starfish wrapped around an unprotected hand. Here, the starfish will be placed away from its natural surroundings on the coral sand. At the tip of each arm, there are sensory organs. They look like longer, thinner tube feet. They're used to decide which way to move. And when it does, it moves fast towards the darker mass of the nearest head of coral. In the lab, a crown of thorns is dissected to investigate its reproductive capacity. This is a female in the breeding season. The gonads containing the eggs are being taken out for study, and soon they fill the dish on the right. This is the egg mass. From this, each year, one starfish is capable of producing 20 million eggs. Harvey Reef, that chosen for the detailed study, is not the most beautiful. Indeed, it shows many signs of damage by the starfish. A sea anemone and its attendants live where hard coral is dead. A jellyfish floats overhead. These are all animals, cousins of the coral itself. Stephen Head.
Corals are fairly definitely an animal. They're quite closely related to sea anemones and jellyfish as well. The principal difference is that it's got a skeleton, though. It has a skeleton of chalk, almost exactly the same as chalk, which the animal itself sits on. It's an external skeleton. This is a fairly simple one. This is Fungia, the mushroom coral. If you can imagine something very like a sea anemone sitting on this, and at night, when the tentacles come out, they'd be waving above it rather like this. This is a fairly straightforward example. Other corals are built up by combinations of these animals, which are called polyps. And you can see it sort of happening as a series. You can take one like this, which forms very large rounded colonies in fairly shallow water. And you can see the polyps themselves sit in quite similar calyces in the rock. But as the thing's grown, it started off as one polyp here, and it's split up, and you can see it splits here and here, like this. Until you end up with, in some cases, a colony of this particular type from one polyp, which is maybe 6 feet across, really very big. If you can imagine this keeping together, growing together at the top, you end up with a pattern rather like this, where the whole thing is very solid and compact, very strong. The polyps are much smaller, but they're basically much the same sort of animal lives in them. They're much smaller and they're very closely tied together. So this, as a result, is quite a heavy piece of rock. It's very solid. You then move on to the stag's horn type of coral. This is a cropper, the one I'm personally particularly interested in. Here the polyps are arranged on axial rods, which come out just like fingers, really. If you can imagine these coming closer together, you get into a form which is a little bit more bush-like, and this is rather a pretty one. And here the polyps towards the tip of the bush have got a rather nice violet tinge to them. Further compressing, you get perhaps slightly more often in deeper water. You have the fingers growing together, and you end up with what we call a table morphology, a table form. This is also a cropper, and the thing will be fixed onto the reef more or less here and sit out like this in a bracket or a small table.
To see how fast coral can grow, we can look here. This was an Italian munitions ship, the Umbria, scuttled in 1941 to prevent it falling into the hands of the British. All the coral here is new. It might have been slow to gain a first foothold on the rusty, paint-encrusted metal, but the largest corals show at a very minimum how much growth can take place in a period of 30 years.
The hydrographic department of the Navy can tell us when the ship went down, usually, and we can then study the amount of coral regeneration which has taken place. Indeed, this coast is so strewn with wrecks, a demonstration of the dangers of coral, that you can almost find a wreck for whatever date you want to check from. And we can see, in fact, the rate of coral growth is amazingly fast in this region, which is obviously pretty favorable to it.
Night is the time to see coral at its best. At night, the animals that need light to search out their food are gone. Many of the fish retreat into crevices and caves under the coral. While this sea urchin, a slate pencil, has come out of hiding. And here a basket star, a night creature which folds in the light of a false dawn. While the animal tissue of the brain coral forms a live and glistening surface to its rocky home as the polyps emerge to take drifting microorganisms from the water. In the study of coral, there may be direct benefits to man.
There are certain possibilities which you might regard as advanced technological possibilities coming straight from coral reefs. A good example is the recent interest in looking for new drugs among coral reef animals. Now, one of the reasons why this is very possible is that coral reefs usually have on them such an immense variety of animals that just by pottering around on a coral reef, the chances of finding an animal which contains some interesting biochemical which may have useful pharmacological effects is really quite great. You may actually discover a new drug. On the other hand, you may find that a particular common coral reef animal is the best source of such a drug.
But the most obvious benefit of coral? With their aqualungs and snorkels and underwater cameras, tourists are welcome to the Sudan. These are Germans, and they could provide an important source of currency to a country that really needs it. But for once, the scientists had got here before these tourists to establish what would damage the coral and what will protect it as an asset for the future. And they've converted local boat owners into avowed conservationists.
The tourists are coming and cutting the corals for beauty, just to take at their homes. But they don't realize they are destroying the area. We have to push them all the time to stop, killing fish or cutting corals and so on.
The Germans have made themselves welcome. For the most part, they politely do as they are asked, which is something of a relief to those who remember an earlier and fortunately smaller invasion by Italian tourists, who arrived armed to the teeth with spear guns. For the unpracticed spear fisherman, it's the gentlest, the most approachable fish that are easiest to kill. He could be killing the very fish that protects the coral from the Crown of Thorns. Besides unwitting man and the starfish itself, coral already has enemies enough. The parrotfish is one. It grazes as though this were a plant, scraping the surface off the coral. Its cousin, the donkeyfish, is not content with scraping. The massive hump behind its head is used to smash the coral down. It excretes the ground-up remains to create the coral sand. In contrast, the Crown of Thorns leaves the coral standing, a clean, white, and perfect skeleton. But to discover the true dynamics of growth, destruction, and regeneration, you need to know what's there to start with. At top speed, they launch an aerial survey. It carries a camera precariously up to 200 feet and looks down onto the reef, the platform, and to deep water again. The next version could be launched from a standing start. This time the airborne camera took still photographs of the reefs. This is a top view of Red Beacon Lighthouse, and from 500 feet, the whole of Red Beacon Reef. And this is the end of the reef where the platform now stands. From such pictures as these, a map of each region can be built up and then surveyed in detail. For this, a 50-foot launch was hired from the Sudanese Navy for an expedition that took them along most of the Sudanese coast. The purpose now to obtain a survey against which all subsequent changes in the coral may be judged. There are many reefs, but this was a summer expedition with plenty of manpower. And trading on their knowledge of where the coral was, they took the rubber boats at top speed to the point of search. 3 divers advancing in line along a reef. This was a method which they soon discovered didn't work too well. If you have to look in every crevice, it takes a long time to cover the ground, and very often they're duplicating their search. It's also more effective to look not for the starfish itself, as this would generally be hidden by day, but for the white patch of coral which shows that a starfish is near. They record the estimated number of starfish in a given length of reef. By the end of the day, they might have found 3 or 30. It's easy enough to recognize the difference between live coral and that which has just been attacked. Newly predated coral is clean and white, but within weeks the evidence fades as green algae settle on the dead coral, and soon it looks like this. Was this killed by the starfish? If so, it's impossible to say when. To know when, you need frequent surveys.
No, that's fine, I think. Okay, yep. Okay, just watch the edge there, it's sticking out the depth.
This was their most effective survey method.
Bit of a fight. That's a predation.
A single diver rides shark bait, as they call it, and delivers a running commentary to the boat above.
How many patches did you say reach? Over.
4, I can think, probably.
The diver is under strict instructions to keep talking. This will protect his lungs if he goes deep and then comes up again too fast. He describes the coral community, its type and its condition, as well as watching out for the occasional patch of predated coral.
The fairly rich coral just near the surface of the top of the coral.
For 2 summers, as they dived and swam through the green algal haze, they found so little damage, it seemed that their original objective, to study the Crown of Thorns in balance, would fail through lack of enough starfish to study. But then the picture dramatically changed. They found a reef that was devastated, crawling with starfish.
In 1970, when we found about 3,000 starfish, there was a spontaneous reaction from our well-trained scientists who immediately called it plague reef. It was only the following year that the term was discreetly dropped.
In fact, within a few months they had found yet another plague, as anyone could be excused for calling this. Seen by night when they come out to feed, their numbers and the damage that they cause are indeed dramatic. But the proper scientific term, one that doesn't prejudge what is happening here in emotional terms, is an aggregation. The discovery of these aggregations in 1970 was a turning point. Did this spell impending disaster for the Red Sea too? Or would it diminish naturally? Was this a natural part of a more complex cycle which was still balanced, but in the longer term of tens or perhaps even hundreds of years? Certainly when they returned the following year, they saw destruction, but with it a reduced population of starfish. Peter Vine, who has seen such aggregations in many parts of the world, recalls how they were first reported in Australia.
The early reports suggested that Crown of Thorns was totally destroying the reefs, and the press rather latched on to the idea that the reefs were going to disappear. My own interpretation is that there were many more skin divers. They knew how to find the starfish, and they knew that it had news value. And so every group of skin divers that found 2 or 3 starfish, or perhaps 20 or 30 aggregated in a small place, reported this to the press. The press were always interested, and so there were more and more stories going out about the spread of Acanthaster, whereas in fact it was a spread of reports.
In Australia, it was the loss of this animal, the Triton shellfish, that had provided the first plausible explanation for the plague. Its beautiful shell, which can reach 18 inches long, is worth many pounds to a collector. So, not surprisingly, divers had been bringing them up to sell them. The divers knew nothing of the Crown of Thorns, or that this shellfish can trap and eat it, so protecting the coral. In the aquarium, the Triton attacks with deadly efficiency. The sole of its foot has trapped an arm of the starfish against the glass. It's cut into the upper surface of the arm and penetrated through to consume the stomach and gonads. Soon the starfish, ripped open and gutted through this hole along the arm, is dead. In all, it's a remarkably efficient piece of specialization. But even if their shells weren't collected, would there be enough tritons to control an aggregation of starfish like the one that was here in 1970?
Here in the Sudanese Red Sea, our extensive searches have demonstrated, I think, reasonably clearly that it's an uncommon species, and it's not possible that there are enough of this very interesting, very attractive shellfish to control the starfish. And we can speak with some authority on this, because the native Sudanese collect all the shells that they can find, which are really rather attractive and saleable. And this particular one, the Triton, is collected, and they don't find very many of them.
If it's not shell collectors, then maybe it's pollution. Pollution of the seas has been suggested as the cause of the Australian and Pacific plagues, so pollution levels are measured here too.
Did you get yours on okay? Did you get it on okay?
One obvious suspect is the Port Sudan oil refinery. It was checked and found to be clean. Another possible source is the shipping that comes into Port Sudan. To check that, The water here is to be compared with that of other inlets along the coast, natural harbours that have no shipping. They take samples and put down plates to check for solids that settle out of the water. But so far, the most important result of this study is again negative. As an explanation for the starfish plagues in the past, or in other parts of the world, pollution can't be ruled out. But there's no pollution here and now. Behind the telegraph office in Port Sudan itself is the lab. And here the group have been working on some theories of their own. Over breakfast, they plan their working day. In this sort of job, there are no set hours, so everyone is free to work from now until night, 7 days a week. The team includes 2 young married couples. The wives, not biologists, soften the still strong expeditionary feeling. Each scientist has his own ideas, but it's the job of the scientific leader to coordinate aims, to decide which of their lines of research shows promise of practical results. It's a fact of life that even the modest funds of a group like this do depend on finding real answers to practical and not just academic questions. One way they've been looking at the problem is to compare the starfish with other related species. Crawling over glass, this is a sea urchin. And here's another. Several years of patient study has led the team to a much clearer understanding of the way of life of many of Acanthaster's closest cousins. But the important question is, do these animals have population explosions like the Crown of Thorns?
By looking at the populations of both Acanthaster and of other urchins and starfishes, you might find one urchin which shows a population increase which very much parallels that of Acanthaster, and then you can then say, well, there must be something similar in the ecology or life cycle of these two animals, such that the same factor is affecting them both. And then by looking at this, you can then attempt to work out exactly what this factor is.
But so far, whether with starfish or sea urchin, no such common factor has been found. These animals are interesting in themselves, but the team has seen no cause, natural or man-made, that produces a population explosion in one of these other curious animals at the same time as the Crown of Thorns. But here's an experiment that did pay off. In order to observe its movement hour by hour, a label is anchored to a spine. This will grip firmly enough to stay on for a few days, during which time its movements can be followed by relays of divers. For ease of reporting, this one is called Linda. This technique was used on an aggregation in which many individuals were tagged. Observation went on round the clock. This experiment was to be one of the most crucial in the whole 5 years' work. It was to lead directly to a theory of how small aggregations of dozens or even hundreds could be formed quite naturally without any need to blame them on shell collectors or pollution.
The really interesting thing that came out of this group of observations was that at the beginning of the night, you probably had 10, 15 starfish, some hiding under a large coral colony, which most of them appear to be predating, a few more scattered around under other, very close by, small coral heads. Then, just after sunset, you found that 1 or 2 began to move, and they moved fairly directly, in fact, up onto the coral colony which was being eaten. And then, gradually, the others began to move, and we were extraordinarily impressed by 2 factors about this process. One was that every animal within the area seemed to know exactly where this coral colony was. It went more or less straight to it, and having got to it, what they all did was try to feed on exactly the same place, and so you had 10 or 15 starfish sort of scrumming, exactly like a rugger scrum, all piling in 2 or 3 deep within the area where eating was currently taking place. Now this in itself seems to suggest either 2 things, either some sort of attraction towards this coral colony for some reason, or else learning by the starfish of exactly where the coral colony was. And it seemed on the whole that the attraction idea was much more likely.
To test this theory of attraction to starfish that are feeding, they worked their way through a whole list of experiments, some in the lab or on boats, some like this on the seabed. Most of their early ideas didn't work too well. They lost one whole series of results because they forgot that a crown of thorns on sand might be attracted towards the nearest patch of dark coral. They kept on trying. Rick Moore.
This piece of apparatus, which we call a Y-maze, demonstrates the ability of starfish to detect the difference between two streams of water, one containing an attractant, which is likely to be related to their feeding activity, and the other none. And this is a reasonable model of the way in which they behave in their food searching activities in the wild.
The experiment is carried out by night. In one branch of the Y there's no starfish, while in the other is the starfish that's been set to feed on coral. Seawater is fed down both branches and will carry any chemical that's released down the arms of the Y and directly onto the starfish in the tank at this end. And already several of them are moving upstream. They're heading up the central channel towards the junction where they must make their choice to go left or right. A few minutes later, the third starfish has reached the junction, and the first two have chosen the path that will lead them towards the feeding starfish. From night to night, the conditions are varied. Sometimes coral is put in the other channel, sometimes starfish that aren't feeding. But the results are clear. The starfish are most attracted by other starfish feeding. In order to try to find out more about the chemicals involved, a biochemist has now joined the team in the Sudan. Here, seawater taken from close to the stomach of a feeding starfish passes down through a column which will separate out organic molecules of different sizes. Each fraction in turn is applied to the sensory tubes at the tip of an arm. Most have no effect, but one does produce a reaction. Here, the starfish can sense, it can taste something in the water. This provides more evidence for the group's theory of how aggregations can form quite naturally on a reef.
The idea roughly we proposed is that when a certain density of the starfish is reached, they're able to detect the byproducts coming from the coral as it's predated. Other starfish from around can detect this, and then they then home in on this source of digestive byproducts produced by other starfish. And this obviously tends to bring the starfish together. And in this way, it seems that we think you can probably get, when you have a population of perhaps 40 or so animals in a kilometre of reef, you can get them concentrated in a very small area of just 100 yards or less. Now, the idea from this stage is that what happens is that once this critical aggregation has occurred, a number of other factors come into play. For example, firstly, we think that the efficiency of reproduction is greatly increased.
Scientists working in the Pacific have suggested that reproductive efficiency may play an important part in creating starfish plagues. The question is, what happens to those 20 million eggs? For natural balance, only 2 from a whole lifetime of egg production should survive. A slight reduction in this vast mortality at some early point in the life cycle could result in not 2, but hundreds or thousands of starfish. One way to find out what causes life or death is to breed Acanthaster in the lab. From a female that's ready to breed, this is part of the egg mass, the organ that produces the eggs. But what will trigger their release? If the chemical message to release eggs can be identified, this too could perhaps be used in some way to control the reproduction of the animal. In this case, the message comes from the nervous system, from the nerve along the arm of the starfish. Barely visible to the eye, the tiny fiber is found. Now an extract is made from it, and the releasing factor is dripped onto the egg mass. Starfish in the Red Sea are capable of producing eggs for over 6 months of the year, a much longer breeding season than has been reported in the Pacific. And now, from a male starfish, sperm will be added to the eggs. In this jar, the eggs will stand a better chance of fertilization than they would in the sea, where few of the 20 million eggs that are released can have much hope of meeting active sperm. On the reef, in summer, microscopic plants called algae drift above the coral, and the starfish larvae live among them, feeding on the floating plant life. They live in a nutrient soup until it's time to settle, to find a place to hide and turn into a true starfish.
When the larvae are ready to settle, they swim down towards the coral. If one of them comes close to the living polyp of a coral, one of the tentacles of the polyp shoots out a mucus thread which attaches to the larva and pulls in the larva towards its mouth. And the tentacles retract and engulfing the larva, and the larva is eventually eaten.
But if they're lucky, they land on dead coral, coral eaten out by an earlier generation of starfish. And now, like Goldilocks looking for a comfortable bed, they search for a hole that's not too big and not too small. Few corals provide a hole of the ideal size. In the laboratory, a smooth surface was drilled out to discover what is a suitable size. These, it proves, are just right for the larva, which enters the tiny crevice to await metamorphosis. On the reef, this dead coral has holes that are perfect. It's a type of acropora. Another is turbinaria, and both of these are corals that the full-grown starfish like to eat. After 2 days, the tiny starfish emerges. But now there's a gap in our knowledge. From this, still smaller than a pinhead, to the grown starfish that are found on the reef, the numbers killed and by what are a mystery. But beyond this gap, the life cycle of the adult can again be studied by following marked animals. For long-term marking, they count the arms and then, using a different code for each animal, they clip the spines in a way that will identify every individual. It's been difficult to find a method of marking that will last, but clipped starfish have been found again after 6 months. They may have fed in one place like this, or they may have traveled half a mile in a week. They can move over surprising distances. The team is gradually putting together a picture of a life cycle in which there are many events which could emphasize the effect of the natural aggregations. One possible conclusion is this.
If they can be interpreted as natural events, there is no cause for concern regarding the geological life of coral reefs. Over a period of 50 or maybe 100 years, one would expect a reef which has been devastated by Acanthaster to redevelop its living coral and to become a flourishing reef again. A visit to many different reefs as a skin diver will always show that some reefs are have flourishing coral growths, others appear to be rather poor in coral growth, others more or less dead. And so there is at present this natural distribution of richness of coral growth.
The natural causes theory. But Christopher Rhodes sees a way in which the presence of man could have been a direct cause of one aggregation they've studied.
The age of the 3,000 starfish we found here in 1970 precisely corresponded with the one point in 10 years' records of the shipping usage of this reef. The one point when the canal was closed in 1967, these ships were here anchored at the height of the starfish breeding season. This certainly seems to suggest that somehow a mechanism operated which resulted in an abnormal number of starfish successfully getting through to maturity.
On this subject, the group itself reflects the division of opinion among scientists throughout the world.
Well, it's extremely difficult to say. There really isn't any concrete evidence either way, to my opinion. But once the population of the starfish reaches a certain level, it's liable to snowball very rapidly. Acanthaster itself, the level of its populations, allows it to be a very sensitive indicator of what's happening in the environment. Therefore, small natural changes may lead to large increases in population, but equally, if man in any way can produce a change which is just as large, then he's going to be responsible for that.
Whether by man's intervention or not, an aggregation on a reef like this could reach the proportions of a plague. Is there anything that man can do to stop it? Here's one possibility. For 2 years, the team experimented with the painted shrimp. From the Max Planck Institute in Germany had come a suggestion that this tiny animal could be imported to infested reefs and persuaded to attack and kill the Crown of Thorns, as it certainly does with other smaller starfish. Would it do the job? Was this the answer?
We decided, in fact, that so much had been suggested and so little demonstrated or proved in the field that it was well worth our while to go to considerable trouble to send people to East Africa, to Mombasa, and for 2 years, for 2 seasons, in 1971 and 72, we had people collecting shrimps and bringing them back personally all the way through to Port Sudan. We conducted a long series of experiments and we demonstrated that the painted shrimp is really not capable of harming an adult Acanthaster, Crown of Thorns starfish.
As a natural enemy, this fish looked a very much better prospect. It does live in the same area, it grows to 8 feet long, and a Sudanese diver had reported it actually attacking the starfish. After weeks of patient stalking, Christopher Rhodes managed to capture these pictures of it, the Napoleon fish.
The problem of watching it is that it's so timid. It's very difficult to approach it. It's exceedingly difficult to see precisely what it eats. So we have to go at it, as it were, back to front. We have to watch the fish market and collect the guts.
This fish has the misfortune to be excellent eating, and if it were to prove important in the natural control of the Crown of Thorns, it would present a problem in conservation. It fetches a very good price in the market. This costs several pounds. Here the whole fish was bought. The rest was to provide an evening meal, but generally just the gut is brought in from the market. A large number have now been dissected to show that the Napoleon fish eats shellfish and some sea urchins, but the Crown of Thorns can play only a minor part in its diet. It was while Rhodes was filming a distant Napoleon fish that another possible predator crossed his line of sight. A shark. The shark is a scavenger, and from Australia there have been reports of it taking the Crown of Thorns. But the shark is more of interest as a threat to the safety of the diver. An examination of its stomach contents suggests that it's not much threat to the Crown of Thorns. But the fact remains that on this reef, an aggregation has died away quite naturally. If it wasn't cut down by any of the obvious predators, what did kill it off? Suddenly, a chance observation found one answer. One of the team saw this fish attacking a Crown of Thorns. He had a camera handy, and this was the first evidence of attack by a triggerfish. And the starfish is dead. Triggerfish are now being studied in the lab.
This is one of the smaller species of triggerfish found in this area, but we can see fairly easily, using this specimen, exactly why the triggerfish get their name. They have this very large dorsal spine, which when they go into a hole for protection, they're able to jam up in an upright position, and this means that any other fish trying to pull them out is unable to do so. And the other group of fish in which we've been interested are the puffers, and these have an equally obvious reason for their name, and this is that when they're irritated by some potential enemy, they're able to take in water and blow themselves up rather like a balloon, and thus prevent themselves from being swallowed.
It was a puffer, a bigger cousin of this fish, that was to provide even better evidence, evidence that was to raise it to equal first on the new list of starfish predators. The puffer is a friendly fish, but with strong, sharp teeth. The importance of the puffer's teeth was discovered by accident. It was during one of the experiments with the caged starfish on the seabed that things started to go wrong, or so it seemed. Something was attacking the starfish in the night. One of the divers was Adrian Sanders.
About the end of September, we came across starfish one morning that had been completely attacked, were killed, and there was a hole in the cage about 4 inches across, showing that something had been in there, attacked them, and then gone out before sunrise when we went down there. The following morning, we found a pufferfish, small pufferfish, swimming around the cage, and it seemed reasonable to assume that it was showing so much interest in the starfish in the cage, or that it couldn't reach them, that this could in fact be a predator. And this sort of made us start thinking, so we'll catch it, start feeding behavioural experiments, and see if it will feed on Acanthaster.
Catching the puffer was easy. Inside the cage, now with stronger wire, it has plenty of room to swim around. And now it's found the starfish. It turned out that the puffer had a systematic method of attack.
Well, they approach basically on two arms and just crop the ends down, and once they've got about halfway down two arms, the two adjacent arms will be exposed, will be free of spines. So once these two middle arms are removed, you have two naked areas. The pufferfish approaches from the side again and just goes around the base of all the arms, removing them. And it's this area that contains the gonads and stomach.
In a mixed diet including other starfish and sea urchins, this fish will eat 2 Acanthaster a week. There are some 5 puffers around this one small reef, and along with the triggers, this should be enough to explain what happened to the 1,500 Crown of Thorns that were here in 1970. The problem now was solved, if the behavior of the fish in the cage was anything to go by. But here again, they had a piece of luck.
Absolutely remarkably, the following morning, we found a larger one outside the cage, and it seemed to be almost devoted to the one inside the cage, which was very useful because we had the natural control to any experiments we were doing, because the one in the cage might have been behaving very unnaturally, but the free one would take anything in its own environment, so it was a natural control.
And since then they've found yet another species of trigger that will eat the starfish, and his method of attack is to turn the animal over to go directly to the stomach and gonads. This reef is very well protected by the natural enemies of the starfish. But suppose an aggregation were to grow to the stage where even these couldn't deal with the numbers involved. Should man then intervene to restore the balance?
If corals are being destroyed by a natural event, in this case aggregations of Crown of Thorns starfish, I think it's reasonable to allow this event to continue, not to interfere. There have been many cases where man has interfered with what he might even recognize as natural events because they're not to his advantage, and he's disturbed the environment more than the original event disturbed it. This may be so in the case of Crown of Thorns.
But those who ask for more drastic action point out that a forest fire is a natural event, and so is a plague of locusts, and we do try to control these.
I think probably once they get beyond a certain size, steps should be taken to control them. That might be, say, 1,000 or 2,000, something like that. The sort of numbers which a couple of men could collect within a few months. And that's within areas such as marine parks, very attractive coral areas close to human habitation where people are likely to come out from to look at the reefs. And a point about that is that I think we're learning increasingly the sort of areas in which these aggregations are likely to form. They're not likely to occur anywhere. They occur in areas of very rich coral growth where you've not got too much exposure, where it's not too stagnant. And if areas such as these were provided with wardens, I think it's very feasible that by regularly checking at a site, such a control would be very easily accomplished.
One method of control is to collect the starfish. Here the team demonstrates a method that's been used successfully in the Pacific. With a bounty of 10 or 15 cents a time, many thousands were taken from the infested reefs off Guam. A problem is that even in plague conditions, during the day the starfish disperse and hide, and it's a difficult and slow job to find them all. If you can find them, another method that's been used is to inject the starfish with something that'll kill them on the spot. If work like this, and the research it depends on, can be done effectively, it may save not only the reef, but also the livelihood of the people around it.
It's leading towards, really, the salvation of small communities. And what we're really trying to do is to help them recognize critical species of fish, which are predators, to make sure that they don't accidentally get turned, as the pufferfish does, into a lampshade rather too frequently. We're also trying to enable those who go out to catch the starfish when there are obviously too many about, to get them densely aggregated. And this is really why we're trying so very hard to find an attractant. We think it occurs naturally, as I've mentioned. We are now trying very hard to isolate the attractant and to use it to cause the starfish against other inclinations to come densely together so that a few people can in a short time deal with a very large number of starfish.